this will be the last time I do this course. I'm retiring from teaching next summer, and that's why they're taping me so that this will go down in posterity. Two of the students I had last year. Let me tell you a little bit about the course. There will be 4 examinations. Each one will count 100 points. And your grade will be based on those 4 examinations. Now, one of the things I do is I'll take the lowest of the 4 and I downweight that test by half. All right, so especially if a lot of students don't know what to expect when they first take a professor, especially for the first exam. And so this is a bit of protection about that. If you ask the students who've taken it before, I'm sure they will agree the best way to do well in here is to read the study guide. I don't know if I've posted it yet, but I will have it by the end of the week. And what it will contain, so each study guide will have 21 questions, and the exam will consist of 6 of those, of which you have to answer 5. All right, so if you really want to start working on that as I go through the lectures rather than try and wait till the last minute to put it all together. The other thing, if you have questions, if there are questions in there that you're not sure how to answer, I'll be happy to help you out. But if you write me an email saying, can you give me the answers to all 21 questions, I will not respond. All right? Other thing about this course, I don't want to see phones. Phones are no-no. And if mine goes off, which it invariably does once during the semester, I will apologize. But I'm too old to figure out how to put it on vibrate. But nobody calls me anyway, so don't worry. Any questions about the evaluation? So 4 exams. One of them is downweighted by half, and that's the basis of your grade. Yeah.
About how long should each answer be in the exam?
I'm reluctant to ask. So every now and then, some student will come in and write 4 sentences and get full credit. They got to be really good 4 sentences. So if in doubt, write more. So I would say 1 or 2 pages of the blue book is almost always sufficient. In some cases, you can get by with less than that, though I don't advise it. All right? Any other questions about the exam? All right, well, why don't we get started then? I want to start with an experiment with you. What just happened?
I see.
Yeah, what did you just see?
He caught it and there was a noise.
All right, how come he's the only one who reached out his hand to catch it?
Threw it to him.
Because he could tell it was coming at him. He was quite surprised by it. You could see that in the expression on his face, but nonetheless, he got his hand out and was able to catch it. If you could figure out how to do that, to get a machine to do that, you'd probably make a lot of money. All right? So what this course is about is to understand how we obtain knowledge about the external environment. All right? That's why I refer to it as high-level vision. Right? We're not concerned, though today I'll talk about the structure of the eye. So we'll do a little bit of basic optics at the beginning of the course. But for the most part, what we're going to try and do is figure out how it is that patterns of stimulation inform us about actions that we want to take, about things that we want to recognize, etc. And that's what this whole course, for the most part, is about. Now, I should warn you in advance, this is not going to be like most courses that you've taken before. We'll have several lectures on the history of art. Why do you think I'd want to do that?
Art history is awesome.
Right? Because artists can tell us a lot, right? Artists represent the world. And most of the early psychology of the perception was actually done by artists in the Italian Renaissance. There are certain areas where we still haven't gotten much past what they knew about. We're also going to talk a good deal about the history of mathematics. Right? We're not going to do any math in the course. You're not going to have to memorize any equations. But even the engineers who are in the course are going to learn some aspects of math that I promise you were unaware of until you've taken this course, like the history of geometry, for example. So let's unpack again what happened that makes this such an interesting phenomenon of him being able to catch the light pointer. All right, so we start, when we're talking about perception, about the structure of the environment. This is the thing I want to know about. I could care less what's on the back of my eye, right? What I care about is that I see in some of the desks there are students sitting and some of them are not, right? Some of the students are males, some of the students are females. Students are different nationalities, ethnic groups, all of these things I can see visually. And the question is, how is it that I know that? Now, if we didn't know a priori that we can make these judgments, so I can toss an object to you and I have enough confidence in my aim that it will get to a place where you can catch it, and the rest of the people in the room are nonplussed, right? You didn't have to worry. You knew it wasn't coming at your head, but he did, and he knew it, and you knew it. All right? And so the question is, how do we know that, and why is it a hard problem? Well, the reason it's hard is because the thing that we're trying to know, the structure of the environment, we don't have direct access to. Right? The environment's not touching me. Right? The way we come to know about it is that light comes from the fixtures up there, comes in through the windows. Light bounces around off the rooms, it bounces off surfaces in the environment, and eventually some of it gets into my eyeball. All right, so the first thing we've done in the act of seeing is that the structure of the physical environment gets transformed into a complex pattern of light in the back of our eye. Now, some of you who are engineering spend a lot of time trying to figure out what that pattern is so you can get robots to do interesting things. Humans do it naturally. All right, so you have the structure of light. This is the immediate stimulus to our vision. And then depending on how you think about things, how many neuroscientists we have in the course? All right, got a big block of neuroscience. So if you're a neuroscientist, you're going to probably think about the physiological processes, how the light is affecting the brain. That's one way you can think about perception. Another way is through computational algorithms. Think about it as an engineer might, right? Where you're performing some computations on that pattern of light to try and make estimates of what's going on in the world. You could exploit prior knowledge about the world, though that's largely overrated, except in cases where the knowledge is very robust and very general. You know, like objects generally are in contact with you in the ground. So from the optical projection you see, right, I could be Professor Todd, 6'2", standing on the ground, or I could be some Lilliputian Professor Todd who's suspended in midair and projects to exactly the same pattern on the back of your eye. But at the end of the day, a miracle occurs, and somehow we're able to figure out from the pattern of light what the environment is. Now, we do not do this perfectly. Our judgments about the environment, we make a lot of errors. They tend not to be damaging errors. We know enough about the environment that we can survive, right? We can pick things up, we can navigate to points in space, we can identify our friends, but we're not perfect at identification, we're not perfect at navigation, we're not perfect at any of that stuff. And so one of the things we study in the field is to try and come up with models of how the visual system works. And the models we're interested in are not the kind of model that an engineer wants, where you're doing it perfectly, because we know you can't. But if we want to understand how people do it, we want the model to make the same kind of errors that people make. Okay, so what I'm going to do today, so we're going to spend most of the course thinking about the external environment, but today and Tuesday, we're going to look at the basic front end of the visual system. There are times when you have to know about this. Even though we're going to spend most of our time on higher level functions, I want to spend a little bit of time on early vision, and we'll do that today and Thursday. Oh, by the way, one of the things I should warn you, you all downloaded the syllabus. Well, when you do, it's on Carmen, and on there it will tell you that next Tuesday's class is going to be canceled. I'm going to be off in Europe giving a talk that day, and so I'll be back on Thursday, but I won't be here for next Tuesday. And my law for my classes is I'll cancel one class, but if it's more than one, I'll get somebody to come to sub. So this term, I only have one speaking engagement, and we'll just cancel it. All right, so let's get to the basics. The stimulus for vision is visible electromagnetic radiation, which can be characterized by its wavelength and its intensity. Now, electromagnetic radiation is a huge spectrum. So microwaves, radio waves, all that is part of the electromagnetic spectrum. We're sensitive to a tiny portion of it in the range of 400 to 700 nanometers. So here's the entire spectrum of electromagnetic radiation. And this is the little notch, which is the stimulus for vision. Now, not all visual animals are sensitive to the same range. So, for example, snakes are sensitive up in the infrared range. Bees can detect UV, which allows them to see spots on flowers that humans can't see. Anybody know what electromagnetic energy is in terms of physics?
An electron, a wave, right, that propagates because it's like, doesn't it constantly switch between the direction that the field is going, and then it...
Yeah, you're pretty close. For a long time, it was a mystery. Scientists learned probably in the 1700s that light was a wave because of diffraction experiments. But they also knew that it traveled through space, and they couldn't figure out how that is. So the waves that they knew about in those days, you had to propagate through a medium like waves on water. And so they kept asking the question, well, what the hell is the medium that light rays propagate through? Anybody know the answer to that? There's a word I know you've heard.
Photons? Light particles?
No, that's the light itself. It's not the medium, right? So when people believe that light had to travel through a medium, how many of you heard the term ether before? It's somewhere out in the ether. Have you heard that expression? Ether is this medium that they just invented. They said, well, there has to be some medium that the light can propagate through. So they said, all right, well, we'll just assume that there's a medium out there and we'll call it the ether. All right, and later on they learned that that's not the way light behaves. Light is actually, it's vibrations in electric and magnetic fields. I think that's what you were trying to say. So you have the magnetism is oscillating back and forth, and you have electric charges are oscillating back and forth, and they're at right angles to one another. So that's the way that light propagates. Now there's one kind of light when you go to, how many of you have been to a 3D movie before? All right, in order to see the 3D movie, what do you have to do? You have to put on those stupid looking glasses. Anybody know what those glasses are accomplishing? They're called polarizers. So the way they get stereo is they have one image where the vibrations are going this way, and another image where the vibrations are going that way. And then if you put on the filters, one eye sees one pattern of vibration, the other eye sees the other. Now, it's interesting that those filters only work in a movie theater. And the reason for that is that most surfaces don't preserve polarization. So if I shined a light against the back wall, right, even if the light was polarized, that's what you call all the light is vibrating in the same direction, the directions get scattered when it bounces off. There's only one substance that preserves the polarization, and that's silver. And that's what they coat movie screens with. That's why they call it the silver screen. Right? So those glasses wouldn't do you any good if you were watching in your living room. You really need a silver screen. All right, now if we look at the back of the eye, it has a rather odd structure. So you got a bunch of cells back there. The light is traveling in this direction, and you got these things called ganglion cells, bipolar cells, horizontal cells, and then these things called photoreceptors, and there are two types of those. They're what are called rods and cones, and I'll talk more about that in a second. But now the interesting thing to note is the rods and cones are the things that absorb light so you can see. But notice they're behind all these other cells. Why do you think that evolution would do that? All right, it's got all this junk in front of the receptors. The reason for that, the reason why the rods and cones are the back is that they use up a lot of metabolism, right? And when you use up a lot of metabolism, you need a lot of blood. And this is where the blood comes from on the back of the eye. So the rods and cones are at the very back, so they have a ready supply of blood. And so this is the way it's designed. The light comes in this way. You've got the retinal, the optic nerve that leaves the retina like so. Here are your photoreceptors. And then you have a whole bunch of cells in the middle, and I'll talk more about these today, that transform the input from the receptors into what will eventually become a neural signal traveling up the optic nerve. The pigment epithelium, which is this tissue right adjacent to the rods and cones, it nourishes the receptors, it's non-reflective in humans, and it absorbs stray light. So here's an electron microscopic image of the rods and cones. Yeah?
I have a question about the cells in front of the photoreceptors. Does the light go through those cells, hits the photoreceptors, and then goes back?
No, it gets absorbed by the back. So this absorbs the straight light. The light does pass through here, and these cells are largely transparent, right? So everything's organized here to allow as much light through as you can get, while still having the receptors being near where the blood supply is. This is a very efficient system. It would be hard to design something that's better than this. And it's amazing for a whole bunch of reasons, which I'll talk about as we go along. So here are photographs of the rods and cones. They got those names because the rods are shaped like little rods and the cones are shaped like little cones. The eye has about 120 million rods and about 5 million cones. And we'll see in a second why we have that distribution. If you look at the distribution on the retina, so here we have the back of the eye, and the way we talk about positions on the retina are in terms of degrees of visual angle. All right, so in the very center, we have the fovea, and this is where most of the cones are located, in a very small section near the center of the retina, called the fovea. That's where all your cones are. Your cones are primarily for daylight vision. Your rods are primarily for nighttime vision. And the distribution, so if you look like so, in the fovea, there's only cones, no rods. The rods increase, so they're maximum about 15 degrees out. 20 degrees out on this side because you have a blind spot. This is where the optic nerve leaves the retina. Now, the question I want to ask is, why do you think we have this? So 120 million rods, 5 million cones. Give you a hint. Cones work in daylight vision. Rods work at nighttime. So let's say you're God and you're designing a visual system and you want that system to be sensitive under really low light levels. What would you do? What would you do?
Almost roads. Mostly they're all roads, not no cones.
Right? You'd want lots and lots of receptors that are really sensitive.
Yeah.
Okay? And so you notice, if you look at the counts, there are 120 million rods. All right? So what that means is, the more rods I have, the more likely one of them will absorb a photon. So if I want high sensitivity, right, what I'll have is a lot of elements. But the question is now, why in daylight vision do we get by with so fewer cones? Now there's another aspect of this that's interesting. So there are 5 million cones, 120 million rods, 2 million ganglion cells. So the ganglion cells are the cells that take information from the retina back to the brain. All right, so the light comes in, stimulates the rods and cones. They then excite other cells along the chain, and the last cell in the link is retinal ganglion cells, right? And they have really long axons that send information from the retina all the way back to the middle of the brain. So just on the basis of these numbers, what can you tell me? 5 million cones, 120 million rods, 2 million ganglion cells. So basically everything that's being detected here is being squeezed into 2 million ganglion cells. And the consequence of that is that you've got a lot of convergence. All right, there are lots of receptors that converge on a single ganglion cell. Now let's think about convergence. So let's suppose I'm a retinal ganglion cell and I'm getting input from, say, 20 rods. All right, retinal ganglion cell, got 20 rods. Say the students in the class are rods. Whenever you catch a photon, you holler something. All right, and the ganglion cell counts how many photons have been collected. Now there's a problem with that. So let's say this class is the number of rods that are collecting photons for the one ganglion cell. That would be me. Right? All the ganglion cell knows is somebody out there caught a photon. But I can't tell if it's you or if it's you or if it's you back in the corner. Right? If you have a lot of convergence of rods to retinal ganglion cells, that gives you really a high degree of sensitivity right? Those cells are really sensitive to low light because any one of the rods could detect something and the ganglion cell would know it. But what it costs you is visual acuity. If I can't tell the difference between you and you, that's not a good thing. Now maybe at nighttime I don't care so much. I'm just trying to get any light in there that I can. But at daylight, that's not a good idea. And so what we find is that if you look at the organization of the system, all right, the rods have lots of convergence onto a retinal ganglion cell, and this gives you very high sensitivity to low light. Whereas the cones are mostly one-to-one. The advantage of that is you get high acuity. All right, so it's like this trade-off. You see this a lot in the visual system, right? If I build the system one way, I get high sensitivity to low light, but I lose spatial acuity. If I build the system another way, I get high spatial acuity, but I don't have very good sensitivity to low light. So what did the human visual system do? It does both. And we'll see that a lot. Same thing happens on audition, right? When we do auditory localization, there's a system in the auditory system that only works for high frequencies to localize sounds. There's another system that only works for low frequencies. You put them together, you work at all frequencies. All right, we'll see that over and over again, right? If there's a trade-off that the visual system has to face, it says, all right, we'll have our cake and eat it too. It'll build both systems at the same time. And so that's what we have. The cone system for high acuity in daylight and the rod system for high sensitivity under low illumination. So here's the idea of rod vision, right? You got a bunch of rods that are feeding into one retinal ganglion cell. The stimulation of any one of these rods will get this cell to fire. On the other hand, if we look at the cone system, it's wired like so. So each of the cones has its own retinal ganglion cell. So let's say you only had one unit of excitation that's not enough to set these guys off, right? If you hit a couple of different rods, that would be enough to set this one off. So there's greater convergence for rods than cones onto the ganglion cells. Thus there's greater summation of rod signals and less stimulation per rod is required to obtain a response. Now again, I want you to think of this from an engineering perspective. You've got two goals. You want to be able to see at night, and you want to be able to see during the day with high acuity. So by having a rod system and a cone system together, you can achieve both of those goals at the same time. Right? Now, if you're a purely nocturnal animal, you might not need the cones. If you are exclusively a daylight animal, you might not need the rods. So I don't know of any animals that have cones but not rods. I know some that have rods but not cones, but not the other way around. Any questions about this before I move on? Right? So again, the way to think about this is from an engineering perspective. What's the problem that we're trying to solve, right? I want high sensitivity in darkness, and I want high acuity in bright light. And by having two systems, you're able to get both of those not at the same time. One works in dark and one works in bright light, but you get them both to work. So cone vision can see finer details. So the rod system, let's say I get this pattern of input as opposed to this pattern of input, they look exactly the same as far as this retinal ganglion cell is concerned. It can't tell the difference. It just knows, hey, some of my rods have been stimulated, but I don't know which ones. As opposed to, in the cone system, I get a this pattern of stimulation and this pattern of stimulation is setting off different retinal ganglion cells. So in this case, you can tell the difference between this pattern and that pattern. And that's what's giving you acuity. So let's talk about how we know about all this stuff. Studying the neurophysiology of the visual system has generated a whole lot of Nobel Prizes. We started being able to do this roughly in the 1940s, I guess. People started doing that. Any idea what technological breakthroughs happened in the 40s? So what do you have to do if, let's pick one of a neuroscientists here. What do you have to do if you're going to study the behavior of a single neuron?
Isolated.
I'm sorry?
Isolated.
You have to isolate the neuron, but then what do you have to do? How do you do the recording? You have to stick an electrode inside it. Now, I don't know if any of you knew this, but the first recordings of visual system, they used a preparation called the giant squid. And who are my neuroscientists? Raise your hands. All right, why did they pick giant squids?
The axon is huge, it's like a millimeter wide.
The axon is really wide, it's like a millimeter wide. And in the 1940s, they couldn't get electrodes that are much smaller than that, right? So if you can only get your electrode down to about 1 millimeter, then you need an axon that's a big hunking 1 millimeter wide, and the giant squid axons were perfect for that. But as time went on, right, the technology for making electrodes got finer and finer and finer. And it was much easier to get them to record from, say, monkey cells or cat cells. And so these experiments took off, the ones I'm going to tell you about, in the 1950s and 1960s, and particularly the pioneering work of Hubel and Wiesel, who both won Nobel Prizes for this work. Now, these experiments, I've actually done single cell studies with monkeys. Now, the version I did is much easier than what they did back in the 50s. So nowadays what they have is a computer that sort of optimizes all the right, so you flash stimuli and you keep changing at random till you find things that work, and then you get stuff that's closer to that. So they have very sophisticated ways of sampling the stimulus in order to see what it is that the cell is responding to. So if you think about it for a second, it's a hard problem, right? I've got some cell in the visual system, I shove an electrode in there, and I can measure when the cell fires an action potential. But how do you know what the cell's going to be sensitive to? Right? Where in the retina should you stimulate? What should the structure of the stimulus be? And so in the early days, they would do trial and error. And so in the earliest experiments, they went weeks on end, where they tried and stimulate the cells they were recording from, and nothing was happening. And it turns out one of the famous discoveries of Hubel and Wiesel, they completely by accident, which I'll tell you about in a second. So the idea here is you're recording from some neuron in the cat's brain, and then you shine a light stimulus on the screen, and you can move it around, have the light be of different size, and what you're trying to discover is what are the properties of that stimulus that that cell happens to be looking for. Now, as a general rule, what we'll see as we move through the visual system, right, that the farther back you go, the more complicated the properties that the cells are responding to. So in the earliest part of the optic nerve, they're just responding to little isolated spots, right? By the time you get back, say, into the infratemporal cortex, they want something that's the shape of a hand or a face, much more complicated patterns of stimuli. So one of the most important concepts here is what people refer to as the receptive field. Now, what a receptive field is, it's not a property of a neuron. What it describes is the region of the retina to which a neuron will respond. All right? So let's say, again, I'm recording from a cell, and I shine a light here, nothing happens. I shine a light here, nothing happens. I shine a light here, nothing happens. I shine a light here, and all of a sudden, ding, 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 action potentials start firing like crazy. I move the light back over here, it stops. All right, so the receptive field is the region of the retina that if you stimulate that region, that's what causes the cell to follow. Now, what makes it a little bit complicated is that the receptive field can have positive and negative parts. So the positive parts, when you stimulate those, the action potentials go up. The negative parts, when you stimulate those, the action potential goes down. So here would be an example. In this case, we have a light shining outside the receptive field, and this cell's firing at its background rate. Now, if I move, looks like that demo's not going to work. I don't have flash on here. All right. If I move the light here, the rate of action potentials would go down. If I move it to the center, the rate would go up. Again, if I stimulate just this one, the rate would go down. So what happens is that in the optic nerve, you've got these circularly symmetric receptive fields. They behave like this. So if you stimulate in the center, it excites the cell. And if you stimulate in the periphery, it inhibits the cell. And I don't think this is going to work either. I don't know why they don't have flash stored on these things from the very beginning, but this is better than my last class. The one I just left, the data projector didn't work. So I was forced to do the lecture giving hand signals and write on the blackboard, God forbid, which I used to do back in the day, but you know, in those days I was prepared for it. I wasn't expecting that this morning. So here are the two kinds of receptive fields of the ganglion cells. There's what's called an off-center, that would be this, where you have inhibition in the center, excitation in the surround. This is an on-center, where you have excitation in the center, inhibition in the surround. And so what happens is, let's say you have a small light right here in the center of the receptive field. You get a rate of action potentials like so. As the light gets bigger, when it just fills the center part, the cell fires at its actual maximum rate. When you start impinging on the inhibitory region, it slows down. And then when you fill the entire receptive field, it fires at its background rate. All right, so what these cells are looking for is a light spot against a background, or a dark spot against a light background, depending on which kind of cell it is. And the sizes of these, yeah.
Can you go back one more time? So for the off-center ganglion, you said it would fire action potentials at its highest when it's dark in the surrounds. Is that the positive part of it?
For the off-center, no.
Okay.
Yeah, a dark region in here and bright on the outside makes that cell respond the most. So when you look at these things, where you see a plus, right, you want light there.
Okay.
And where you see a minus, you don't want light. Light in the minus area causes inhibition. Okay?
Thank you.
Now the other thing to note is that the size of these things increases as you move to the periphery. So what that means is that all these early cells are looking at one color against a background. So let's suppose that you're up in Alaska, say, and the sky is white with clouds, and the ground is white with snow. Right? So it's white everywhere. What would happen? How would these cells respond? Anybody?
Just like they normally do.
What would they do?
Just background.
Just background. So it's as if they weren't being stimulated at all. Right? That phenomenon, by the way, is called snow blindness, where people report that they lose their sense of vision altogether. Because if all you have is homogeneous white, you're basically not stimulating the system at all. It wants contrasts. That is called a Ganzfeld, where everything is exactly the same color. And they used to do this. People would put like ping pong, half cut out ping pong balls over their eyes and get like in a sensory deprivation tank. And they report their experience of vision just disappears because they're not getting a stimulus that their brain is set up to analyze. Okay, now remember, I'll give you some examples to try and make this a little bit more intuitive. So what we're looking for is this cell wants a bright spot against a dark background. And let's see if we can see any behavioral effects. This was well known before anybody recorded from neurons. And it was known primarily because of this effect. Which looks brighter, this square or that square? Let's pick on you. Which one looks brighter?
The one on the right.
The one on the right. You sure it's brighter?
I don't think it is, but it looks like it.
You think I'm fooling you?
Yeah.
You've heard rumors about this class. I fool the kids in this class all the time. This is the first example of it. So let's move it out of the background. And now they look the same. Move it back in, and they look like they have different colors. And the reason from that is because of this lateral inhibition. Now here's a very strange effect. Let's pick on you again. Tell me what you see here.
Like little dots in between the squares where they intersect the corner.
And what's causing those dots?
Probably a visual illusion.
Probably a visual illusion. What happens if you look directly at one of the junctions?
It disappears.
It disappears. You only see it when you look away.
Yep.
All right? Anybody have an idea about why that happens? So there's two things we want to explain. Why do the dots appear at the junctions? And why do they disappear when you look right at them? You got a suggestion?
Well, earlier you said that ganglion cells are larger in your periphery, so if you're not fixating on it and you have a large ganglion cell, doesn't that mean you have
Ding, ding, ding. You win a gold star. That's the harder of the two questions. All right? So why do the dots appear at all? So you explain why they disappear when you look at them.
Why would I didn't hear what she said.
She said the cells in the periphery have larger receptive fields than the ones in the fovea. And that is the correct reason why they disappear when they look at it. Now, if you got there, you ought to be able to tell me why the dots appear when you're looking away. All right, well, let's look at an example of this. How much inhibition do I get for a cell that's positioned here? Only in these two regions. All right, whereas a cell positioned here, you're getting inhibition from this, and this. So you get twice as much inhibition here as you do there. That means the cells that are responding are in this region are giving you lower rates of action potentials than the one here. And if that's what's responsible for the color that you see, that explains the why the dots are appearing at the junctions. Now that was the easier of the two questions, right? You got the hard one, which is it doesn't really matter if the receptive fields are small enough that they fit completely within the rows or the vertical rows, whatever they are, fit in the junction. All right, so this is a good example of how we can take this notion of lateral inhibition, see the wiring, the way the cells are behaving in the optic nerve, and you can actually explain perceptual phenomena with that. Now, it turns out that the situation is a lot more complex than what I just told you about. When you get to retinal ganglion cell types, there are several different types and they do different things. So one type, and these are the most common by that's the M cells. No, sorry, the P cells. P cells are the most common by far. They have small receptive fields. The cells themselves are small. They give a sustained response. So what that means is if you turn a light on inside the receptive field, the cell will just keep responding until you turn the light off. The P cells respond to color, and they're about 80% of the retinal ganglion cells. So the vast majority of cells are P cells. They give a sustained response and they respond to color. Now the next class are interesting. They're called M cells. They're much larger than the P cells. They have larger receptive fields. They're transient. So what that means is that when you first turn a light on, you get this burst of response, and then it dwindles down, and then you get another burst when you turn the light off. And these cells are responsible for our sense of motion. They're about 10% of the cells. And then finally, we have these things called K cells, very small. They're also transient. They also respond to color. And people aren't completely sure what these things are doing. I think the best guess, I should be careful here because I'm hardly an expert on this stuff, but last I read about this, the best guess is that they were involved with like adjusting pupil size in bright or dark light. So one of the wonderful things our visual system does is we have dynamic adaptation to different light levels. So when you're in bright light, your pupil shrinks to let less light in. And when things get dark, it opens up wide. So I'm sure you've all been to the optometrist and they put those annoying dilation drops in your eye. You all had that done. And then you've never had that done? Have you ever been to an optometrist?
I wear contacts, glasses.
And they don't dilate your eyes?
And puff stuff in it, like air.
Oh, that's incredible. You should get a different optometrist. They really should. I mean, that's why they look back there to see if there are any unusual growths. I remember the first time I was dilated, it was the guy's looking in my eye, didn't have the best bedside manner. He looks in and he goes, "Oh, that's not good." And but I knew right away what he saw. When I was a little, maybe 12, I was playing baseball. I was playing catcher without a mask, and I got hit right above my eye over here. And in fact, they thought I was going to lose vision in that eye, but fortunately, they got me to the optometrist soon enough that they could save it. There's a big hunking scar on there from that accident. And I figured out right away that that's what it was, so I wasn't too freaked out, even though he was talking cancer. I knew better. Now I go to my optometrist and she looks in there and she says, "Oh, that's so beautiful." And I always say, "You have a bizarre sense of aesthetics." Actually, I may have that scar here. Sometimes show it in class. I guess not. So this is what a P cell looks like. So it's firing its background rate. And now if you turn the light on, whoops, flash is not working. I guess that makes sense. Flash doesn't really exist anymore, does it? They stopped supporting it.
Steve Jobs killed it.
What's that?
Steve Jobs killed Flash with his dying wish. It's like a real thing.
It's a good thing I'm quitting teaching. I got hundreds of Flash demos in my lectures. They just have, I go through this every year. They don't install it unless you ask them to. So here's the idea. If you shine for a P cell response, if this were flash, a light would go on here and the rate of action potentials would go up. You shine it in the periphery, it would go down, and it just stays that way. As opposed to, for an M cell, again, if you, I don't think this is going to work, but I'll try anyway. Oh, it did work. Well, it's not really working, but anyway, you turn the cell on, you turn the light on, you get a burst of activity, it goes back down to its baseline. You turn the cell off, then you get a reduction below the baseline, and it gradually comes back up to its natural rate in the absence of stimulation. Yeah.
Does it mean the P cell detects if it's there, the stimuli is there, and the M cell detects if the stimuli is changed?
Yeah, the M cell is responding to change, and the P cell is responding to the steady state. Later on in the course, I'll tell you about an experiment that I did in my doctoral dissertation. It was based on that. Whereas if you're looking at a screen and a stimulus appears, it just grabs your attention automatically. You get really fast response times. As opposed to, let's suppose you had all possible stimuli up there at the same time, and you turn everything off except one, your response to that is much slower. So what happens in those P cells, they're like an alerting system that when they fire, they just grab your attention. So if a light, say, some other human just spontaneously appeared next to me, right, all of you would instantly stare at it. You can't resist. Whereas if everybody in class disappeared except one of you, you would be probably freaked out by that, but you wouldn't automatically look at the one person who's left. So we'll talk more about that later when we get to attention. I think what I'm going to do is I'll quit now. Next time we'll talk about what happens as you move farther up the visual system, and then we'll be done with our neurophysiological preliminaries, and we'll start talking about attention and perceptual organization and things like that. Any questions before I let you go, either on the lecture or aspects of the course?
There's reading.
Yeah.
Sorry. Go ahead.
What-
Okay, just want to double check. Will the slides be on Carmen?
The slides are all on